Hello everyone, I'm Dr. M and this is a supplemental video to my genetics lecture course. And this video will cover the Mendelian principles of segregation and independent assortment, as well as associated laws of probability that follow from these principles. So when Mendel did his famous P experiments, what he did was he crossed two true breeding lines. And what this means is that these lines, these plant varieties, were homozygous. And he crossed homozygous, or for him, true breeding, because he wasn't totally aware of homozygosity just yet. He crossed true breeding round-seeded plants with true breeding wrinkled seeded plants. That's our parental generation. And he created the F1 generation, our first filial that were all hybrids, and they all had the same phenotype of round seeds, which resemble one of the parents, but not the other one. This demonstrated to him that this trait was not inherited by blending inheritance. Under blending inheritance, we would expect the F1 to be somewhere intermediate between the two parents, but this was not the case here. There are other traits that do seem to be inherited along those lines, but this was not one of them. He then selfed these plants to create the second generation, the F2s, and I guess we should call it the third generation. And he found 3/4 of them were round, but 1/4 was wrinkled. And so he was able to recover this parental phenotype in the P generation back in the F2 generation. So he concluded that there was something that was inherited by this parent that was still present in this individual that was then passed down to this individual. And so he concluded 5 things. First, that a hereditary factor, some factor, produces a trait that is inherited. Second, each individual carries 2 forms of the gene. We now call these alleles. One of these alleles, so our different alleles are causing our different phenotypes. One of these alleles is dominant over the other. This means that in the hybrid, in the heterozygote, the dominant allele masks the phenotype of the recessive allele. During meiosis, alleles segregate with equal probability into gametes. So what this means is that in this individual's gametes, seeds and pollen in this case, can contain a big R or a little r with equal probability. And then at fertilization, gametes also fuse with equal probabilities. And so you're just as likely to get an individual that's big R here, little r, big R here, big R, little r here, or little r here. And so each of these possible combinations between big R and little r are possible. So you would expect each of those to be of equal probability or equal proportion. Now, but what about the 1:2:1 ratio, where we have one homozygote dominant, one recessive homozygote, and then 2 essentially heterozygotes? Well, the 2 heterozygotes are actually not exactly the same, because one heterozygote got their big R from mom, while the other heterozygote got their big R from dad. And so they're both heterozygotes, but the nature of how this individual's parents made them heterozygote is different. So that's the distinction. So that's how we end up with 2 heterozygotes, but phenotypically they're the same. But we can think that maybe genetically they're just, they came to it from different ways. Okay, so that's the principle of segregation. What this is talking about is that during meiosis, during gametogenesis, what this means is that when homologous chromosomes are lining up on this metaphase plate, they are doing so randomly. So here we have two chromosomes. Well, we have 4 chromosomes, but we are diploids. So we have 2 copies of each of our 2 chromosomes. One copy is red, one copy is blue. Our different chromosomes that have totally different genetic material on them are designated by size. So we have a big one and a little one. They're homologous chromosomes because one of them came from mom, one came from dad, and they could have different alleles on them, or they could have the same allele on them. Now, these chromosomes look like an X because they've replicated, and so each chromosome is comprised of 2 molecules that are identical to each other. And these 2 molecules are called sister chromatids. All right, so right now we have homologous chromosomes lining up on the metaphase plate during metaphase 1 of meiosis. And the principle of segregation means that you have an equal probability of the red homologous chromosome of the big chromosome, ending up in the same daughter cell as the red homologous chromosome of the small chromosome. We could get red and red together. You could get blue and red together. You could get red and blue together. You could get blue and red together in those 2 different combinations that will ultimately end up making those 2 types of heterozygotes. Okay, and so what the principle of segregation means is that this is totally random, how chromosomes line up on the metaphase plate at this stage and then also at this stage. Okay, so from a cellular perspective, I wanted to explain where those principles are actually coming from. Of course, Mendel did not realize this, but yeah. So, okay, independent segregation of alleles is true whether or not you have crossing over. So we can think of it as being independent of crossing over. So what this means is that, let's say you have no crossing over, and here we do have some crossing over, where our, what we're looking at is a single homologous pair of chromosomes. This homologous pair has replicated, and it's a heterozygote because we have a big R and a little r. Here we have no crossing over between our homologous chromosomes. Here we do have some crossing over where this little r has switched places with this big R. Okay? So now on our sister chromatids, one has a big R and one has a little r. That's what crossing over does. And as we're going to see in the next slide, okay, let's continue this down through the rest of meiosis. If we have no crossing over, we're still just as equally likely to get a big R in a gamete as a little r in a gamete. If we have crossing over, we are still just as likely to get a big R in a gamete and a little r in a gamete. It's a little bit silly to talk about whether or not crossing over matters for the principle of segregation, just because crossing over only matters when you're talking about more than one gene, as we will see coming up. Now, okay. All right, so what this means, if we have an equal probability of getting different alleles in different gametes, what this means is that we can use basic rules of probability to predict genotypic and phenotypic ratios. So there are two important rules when it comes to calculating probability. There's the addition rule and there's the multiplication rule. The addition rule is applied when you're trying to figure out the probability of an event, a first event or a second event. So if you want to know what the probability is of event X or event Y, you add them. This is the sum of their individual probabilities. So for example, if you wanted to calculate the probability of the Seahawks winning the Super Bowl or the probability of the Patriots winning the Super Bowl, you would add them. And considering that those are the only two teams playing in the Super Bowl, that probability sums to 1 because one of them is going to win. The Seahawks or the Patriots are going to win. That's guaranteed. So that is a probability of equal to 1. If we wanted to understand the probability of two events happening simultaneously, then we multiply their individual probabilities. So to reword that, the probability of X happening and the probability and Y happening is the product of their individual probabilities. So if I wanted to estimate the probability that I would get an A in genetics and an A in organic chemistry, you would have to multiply those probabilities. Mathematically, because probabilities are always expressed in terms of a fraction between 0 and 1, because they're always less than 1. They're either 1 or they're less than 1. The probability of one event or the other event happening is always going to be larger than the probability of them both happening. And intuitively this makes sense. If you flip a coin, the probability of getting heads or tails is 1. This is bigger than the probability of getting heads in one coin toss and tails in the other. Okay? So those are the addition and multiplication rules. Now, just some practice questions. The probability of flipping 2 heads in a row. You can go ahead and pause it and see if you can figure it out, and then I'll come back and give you the answer. Okay, so the probability of flipping 2 heads of a coin in a row is going to be, these are 2 simultaneous events, so we're going to multiply. So the answer is going to be B, 0.25, because we have a 0.5 probability of flipping heads and a 0.5 probability of flipping tails. Or sorry, we have a 0.5 probability of flipping the first head, and then we have a 0.5 probability of flipping the second head. Then both together we multiply, and that's 0.25. Okay, here's another one. The probability of flipping a heads or a tails. So the answer to this one is going to be E, or 1, because you are adding. The probability of flipping a heads is 0.5, plus the probability of flipping a tails is also 0.5, and this comes to 1. And so when you think about it conceptually, if you're going to flip a coin, you will get either heads or tails. Okay, this one's a little bit trickier. The probability of flipping a coin twice and getting a head and a tail. Okay, now there's 2 ways to get a head and a tail. You can flip flip a head first and a tail second, or you can flip a tail first and a head second. And so normally you would have a 0.25 probability. But because there's 2 ways to do this, it actually, you sum those, and then you're going to get 0.5. And so one way to think about this, I will continue talking about this in the next video because I am about to run out of time on the clock.